So, welcome. Welcome to this lecture session, which is beginning of Advanced Microscopy workshop. This workshop is organized within MedGenet project, which is part of twinning program of European Union. So today I have a great opportunity and great pleasure to introduce our two speakers, Michael Stone from United States and Mark Hink from Netherlands. Mark and Michael will guide us through most advanced fluorescence microscopy techniques, and also they will tell us about their applications. So without further ado, let me introduce Michael Stone from University of Santa Cruz. Michael was able to accomplish his BA degree at University of Pennsylvania, as you can see here. And then he went on with his career at University of California, Berkeley. And then after his studies, he joined Harvard University, where he started to use single molecule fluorescence microscopy techniques in order to reveal new things about ribonucleoprotein complex telomerase. Michael, as you can see him in his postdoc years, then moved to University of California, Santa Cruz, where he started his own group, and Michael's team investigates the dynamics, structure, and function telomerase and telomeres. Michael is well recognized in the field of telomerase and telomeres, and I can document it by a real story. When I was visiting the laboratory of Tom Czech at Colorado University, Boulder, Michael visited his group. And during that time, Tom asked Michael to lead a whole lab meeting. So Michael is the only person that I know that was given such a privilege. So I'm really glad that we have Michael here, because I know that he enjoys teaching a lot, and we will have really great opportunity to enjoy him in this workshop. So please show him how enthusiastic we are by big applause.
I see a lot of smiling faces, which is good, and hopefully you'll still be smiling at the end of the hour, okay? Let's see. Over the next 2 days, I'll have a great opportunity to get to know many of you on a more personal level, where we can together enjoy working on the microscopes and learning some of the technical aspects of some of the techniques that we use in my laboratory to study various aspects of molecular function and dynamics, as Dirad mentioned. In my lecture today, I'm going to try to avoid overburdening you with too much technical detail, but rather prefer to focus on more conceptual applications of the types of spectroscopy and microscopies that I've been involved in, both being a student and then ultimately running my own group and helping to develop aspects of this field. I know that my talk in the program talked about studying dynamics of nucleic acid structures using specifically the technique of single molecule Forster resonance energy transfer, or FRET, but my talk will actually involve techniques beyond that specific technique, though we will conclude with some discussion of the FRET technique. And I'll start, for me it was fun to begin to put this together because it was an opportunity for me to let's say, reflect on my own path over the last, I was just speaking with Mark, say 20 years since I've started using single molecule techniques. Hard to imagine it's been that long. But when I started, really as an undergraduate, interested in macromolecular structure and function, this was sort of my starting point, personally. I had read James Watson's book, The Double Helix. I don't know if any of you have ever read this book. You may know James Watson is an interesting character, but I'll leave that for another day. But he obviously made seminal contributions together with Francis Crick in their description of the double helical structure of DNA. And I remember as a young student being enamored by just the notion that we could even describe molecules at this level of detail in terms of their structure, right? Flashing forward, so the title of my talk will, which is a little cryptic at this moment, will become more clear as the talk goes on, is this idea that many of us, when we think of DNA structure, we think of this double helical structure, or so-called B-DNA, B-form DNA. But what I'll tell you about is the fact that DNA is not a static molecule that only exists in this one structure that Watson and Crick described in the 1950s, but rather can take on numerous alternative structural forms under various, let's say, cellular conditions. And we'll talk a little bit about some of these more unusual structures of DNA and how single molecule techniques can help us to study those structures. Okay. Oh. Oh, I realized I have the wrong, we have matching, let me just stick this in here. Okay. That should work. Good. Okay, so this is sort of the mission statement of my research group. We call ourselves the Stone Research Group, or SRG for short. And my reason for putting this up here is that I'm going to focus on single molecule techniques. That's what we're here to discuss. But I think it's important for all of us, particularly students, to think about that for tackling complex biological problems, no one technique is the answer. Okay? And this is something that I was trained on very early on in my career, not to just have a single hammer, to constantly look for different nails, but rather to have diverse tools. Okay? And this is, in fact, how I run my research group. And so we use single molecule biophysics, as I'll describe, but we use a suite of other experimental and computational approaches to attack the problems or to address the problems of the day. And these can range anywhere from more traditional, what I call test tube biochemistry rather than single molecule spectroscopy, to high resolution structure determination and some computational modeling. Okay? And so I think the key to attacking difficult problems in biochemistry or biology more generally is taking a multifaceted approach. Okay? And so I just put that in as a disclaimer because now I'm only going to talk about single molecule work, this blue circle, okay, from here forward. So let's talk about single molecule biophysics. So the way I'm going to structure this lecture is to spend the first 20 minutes or so talking more conceptually about why one might want to do a single molecule experiment. Anyone who has done such an experiment can attest to the fact that very often they're difficult to set up. And so one should have a very good motivation or reason to do such an experiment before diving into this sort of technically challenging endeavor. And so I'm going to talk about some of the unique types of information that one can extract by the act of doing single molecule experiments. And then I'm going to quickly just show you some instances, not of my own work, but of work that has really served as an inspiration for me, where dynamic systems in biology have really been probed using different types of single molecule spectroscopies. And that all seemed to happen. I was lucky in a way that when I began my graduate studies toward the end of the 1990s, there was a lot happening in this field. And the techniques had been sufficiently developed where they were beginning to be applied to some really interesting biological systems. And this was just as I was sort of deciding what it was I was going to work on. And there was a lot of inspiring work. So I thought I would take this opportunity to share some of that with you. And then we'll segue into the main focus of the research component of my lecture, where I'll talk about two different studies, one old and one new, where I personally was involved in studying interesting properties of DNA, which again is what got me into the scientific process to begin with. So you might be wondering what this picture is meant to represent, this idea of a molecular flea circus. So I posed the question, single molecule biophysics, what can we learn about biology or biochemical processes with this notion of a molecular flea circus? So I'd like to get a show of hands. How many of you know what a flea circus is? Not too many, a couple, okay. So a flea circus, I like to make a joke when I talk about this with my young students that before there was, Xbox or PlayStation, people had to entertain themselves some way, right? And so this was sort of an early version of an entertainment system, right? Where you had these macroscopic, but let's call it microscopic for the moment, little bugs, fleas that you cannot see, right? Driving the motion of larger objects that you can see. So a classic example is maybe a small bicycle going across a tightrope, right? And you look at this in the flea circus and the object is moving, but you can't really see what is driving that motion, okay? So what am I talking about? You might think that the jet lag is starting to get to me a little bit right now, which it might be. And so the analogy will become clear as I talk about how you actually have to go about studying single molecules, okay? Because one way or another, if you're going to study the dynamics of a single molecule, whatever that molecule of interest might be, you have to put some sort of tag onto that molecule. And so most typically, that tag either comes in the form of some sort of fluorescence probe, okay, that you attach. So this is a relatively small tag, but that gives you sort of a handle of light in order to actually see the movement or motions of the molecule of interest. Or very often the tags are large plastic beads that you can attach, and these are literally handles with which to pull and manipulate molecules. And so in a sense, you're not actually looking directly at the molecule itself. It's far too small to do that in a dynamic way, at least simply. And there are methods, right? But if you put these macroscopic tags, then you have this idea of a molecular flea circus, right? Where now, through the motions of these objects that you can see, you can then infer, right, what is actually driving the motion, okay? So that's the concept that I mean to put forth with this idea of a molecular flea circus. But first, let's talk about some basic ideas, concepts, with these sort of toy examples, cartoon examples of what types of information are difficult to extract from a beaker or a test tube, but that if you had some way to study at the single molecule level, you could imagine that certain bits of information just fall out of the experiment, okay? And so, for example, one idea you can think about is you might have an enzyme shown here schematically as some yellow object, either a square or a triangle, okay? And imagine that these two enzymes, which are the same enzyme in the sense that they are the same gene product, okay, the same protein, let's say enzyme, but have some sort of unique conformation or unique structural property that confers a unique functional output. So let's just think of that in terms of the rate of enzyme turnover in this particular case. So you might have a slow enzyme and a fast version of that enzyme, okay? And this enzyme catalyzes the conversion of a green substrate into a red product, okay? So you could imagine that if you're studying, the millions and millions of molecules that you typically would be studying in a test tube measurement, then as a function of time, and we're going to come back to this idea of time, okay? Because this is really where, when we talk about dynamics of molecules, you're really talking about how these molecules are behaving as a function of time. It would be very hard to actually extract, not impossible, but hard in certain ways to extract different enzyme rate fluctuations from the ensemble of behaviors because all of these behaviors are not synchronized to one another, okay? However, some of the early single molecule enzymology experiments overcame this challenge by essentially taking an enzyme and sticking it down onto a surface, okay? So now imagine you don't have a whole tube or a beaker full of the enzymes, but you have one, okay? Or maybe several hundred, but you can resolve them individually, okay? So now you have your slow enzyme and your fast enzyme. And if you had some way, say through light microscopy, to measure the turnover of that enzyme, now it becomes almost trivial to say, well, the square is the slow one, okay? And the triangle is a faster turnover version, okay? And indeed, when people first started studying enzymes at this single molecule level, this is precisely what people found, was that products of the very same gene, okay, nominally the same enzyme, the same amino acid sequence, okay, can give rise to quite different enzymological behaviors. And that actually came as a surprise, I think, to a lot of enzymologists in the early days. A name was given to this phenomenon called static disorder. So this idea that a particular molecule, although if you looked at its structure, say in a crystal structure at high resolution, might look identical, somehow could give rise to very different rate behaviors, okay? So that's not terribly, I guess, in retrospect, shocking or surprising at this stage of the game. What I think was most striking, though, is that in similar types of studies, researchers found that a slow enzyme could transiently turn into a fast enzyme. Okay, so now we have this phenomenon, right, where you have essentially an enzyme with a gearbox, right, that can go into a fast gear and then shift down into a slow gear, okay? So now as structural biologists and mechanistic-minded people, we have to think to ourselves, well, what is happening to this molecule that's changing it from a fast turnover to a slow turnover? And these are often not subtle effects. In fact, some of the early studies performed by Sunny Shi at Harvard University demonstrated that a particular enzyme can exhibit rate behaviors over a range of about 5 orders of magnitude. Difference in rate constants, okay? So these are not subtle changes, but can be actually quite dramatic in terms of their variance. And so this idea was called dynamic disorder, this idea of a single molecule to change its rate behavior. And it really is a very nice example of how, as entomologists, we had to change our way of thinking of how these molecules may be behaving. And you could immediately begin to speculate about how these changes in rate behaviors could relate to regulation of enzymes. Sometimes it might be better to go fast and sometimes slow, and so on. Okay, so this is one idea that came out very early from just the ability to look at an individual enzyme turn over its product in real time. Okay? Another classic example is if we use, again, our schematic of a green, so let's say, unfolded structure going to a red folded structure. So let's imagine now we're looking at a protein or an RNA folding pathway, right, which is a common sort of paradigm to study, that might go through some transiently sampled intermediate. Well, again, using our analogy to an ensemble measurement, transient intermediates are very difficult, if not impossible at times, to characterize. And this is, again, due to the asynchrony of all the molecules that are present in that test tube, okay? And so even if you have some tricks, and many people have done this over the years, to synchronize the molecules at the start, slowly as a function of time, these because of the stochastic nature of biological processes and folding processes, the population quickly becomes asynchronized or desynchronized. But again, if we think into our mind's eye and say, well, what if we had the capacity to pluck out of this beaker individual molecules, again, stick them down onto a surface, okay? And instead of watching an entire population evolve from green to red, instead track individual molecule trajectories as a function of time, again, and study, well, okay, this one stayed green to this point, transiently turned into this intermediate before getting to its final so-called native state, right? And in this way, you can easily, again, it's an example of how if you have the capacity to perform such a measurement, some of these complexities immediately fall out of the observation. So you can immediately detect things like these transient intermediates, characterize them, measure their energetic stabilities, and so on. And in many cases, it's been found that intermediates that could not have even been really guessed at in the ensemble type measurement show themselves in the single molecule experiment. So again, this ability to watch the time evolution of an individual molecule, particularly in this case, in situations where you're studying folding processes or unfolding processes, becomes very powerful. Okay, so those are just two obviously very conceptual examples. If we stopped at green and red circles and did nothing else, we'd have a hard time getting funding for our research, right? But it's a good place to start to just be thinking, well, what's the point of all of this, okay? It's all well and good to be able to look at single molecules, but you ought to be able to learn something unique from the measurement, okay? Okay, so now coming to things that are more complicated than red, yellow, and green circles, okay? The cell, okay? So I don't know if any of you are familiar with the artwork of David Goodsell, but you've probably seen pictures like this of his amazing work. And he's a painter who renders cellular images all drawn perfectly to scale, okay? And I always like this as a starting point because we as, I should speak for myself, I as a biophysicist and my students and postdocs very often are studying purified samples in aqueous buffers, right? And we must remember that this is not the natural habitat of these molecules, right? The natural habitat of the molecules is this very crowded confine of a cell. And what we're looking at here, just to orient you, is sort of a cross-section of the nuclear envelope here. We have little nucleosome octamers, core particles wrapping the DNA here. This is a rendition of the nuclear pore complex as mRNA molecules are sort of making their way, exporting out into the cytoplasm to meet up with the machine, the ribosome, to be translated into proteins, right? Some of these proteins are adhered to the endoplasmic reticulum here for proteins that are destined to be either displayed on the cell surface or excreted from the cell. And so, the point here is that the cell is both very crowded, but as I'll show you, is also very dynamic. And so when I'm going to show you now some examples of movements of some of these macromolecular machines. And it's always good to keep in the back of your mind that this is all happening in this environment, which after even studying this for 20 years, still blows my mind, right? That any of this actually works, right? That a molecule can get from one end of the cell to another to trigger some sort of regulatory cascade, for example. And one role that single molecules have to play is sort of to take static images like this nice artistic rendition by David Goodsell and bring them to life in some way to actually understand how these molecules in this sort of crowded environment might actually move and in turn function. So I'm going to give you, I think I included 3 classical examples of single molecule dynamics, again, which are not my work, but served as inspiration for some of my work. Okay. So I would argue that some of the earliest single molecule dynamics measurements were done by these 2 fellows here, Nair and Sackman, who ultimately won the Nobel Prize for their work studying the molecular properties of ion channels. Okay? And they came up with a very clever technique in the early 1970s called patch clamping. It's an electrophysiological technique. And I don't want to go into the details of the individual techniques being used here, but the main point here is that they could use a small glass capillary and pinch off a part of a cellular membrane such that they would have a single ion channel in this little aperture of the capillary, and they could apply a voltage across this membrane with this single ion channel and measure the current passing through, okay? And in doing so, in this sort of patch clamping experiment, you could see that, again, as a function of time, this is a normalized value. I should have had a label here of picoamps. This is the current, basically, that they're measuring. They could actually detect when the channel is closed and essentially no current is passing through that opening. And then transiently, you can see this is on millisecond time scales, the ion channel will open, okay, and show a little characteristic current, which is shown here as this lower potential. And so what you're supposed to take from this line is that basically closed channel goes to open, closes again, goes to open again. And so you're actually seeing in real time an ion channel opening and closing, okay? These experiments, early 1970s again, were really the first single molecule dynamics experiments where we began to understand some of the stochastic properties, the rate properties, how different structural perturbations, say in the form of mutation, affected the opening and closing kinetics of single ion channels, okay? And one thing that you'll notice throughout the rest of my talk, and often some of my colleagues who don't do single molecular spectroscopy joke with me, they say, everything I do looks like this, right? They say everything looks like that, which is true. You'll see that. But of course, it takes one's ability to extrapolate beyond that to understanding what is that molecule that's driving that signal actually behaving and doing. So that's single molecule ion channel recordings from the early 1970s. This experiment here is probably the one experiment that inspired me most to get into single molecule work, because it was one of these jaw-dropping experiments that happened right around the time when I was starting graduate school. So some of you might be familiar with this complex called the F1 ATP synthase. This is the biological machine that basically couples energy from a proton motive force or a proton gradient in order to synthesize ATP in our cells, which, as I hope all of you appreciate, is a very important process for the cell to survive. And through decades and decades of biochemical and biophysical and ultimately high-resolution structural work, models had emerged for this complex, which is basically sort of a donut or a toroidal-shaped hexameric structure that has on its interior the so-called gamma subunit, which is you can think of as sort of an axle sitting on the interior. And there was basically a model put forth that this alpha-beta hexamer, shown from the side, and its gamma subunit on its internal region, that there, as part of the mechanism for synthesizing ATP, that there should be a rotation as the protons are moving through and driving this process. So many people for many years debated, does it rotate or does it not rotate? And you'd be amazed at how many scientists can have strong feelings over such a question. Rotate, not rotate, okay? But it gets at the inner sort of mechanistic function. And so one of my all-time heroes, the late Professor Kinoshita from Japan, he sadly died in a hiking accident. So he apparently was a very avid hiker. I think there are several thousand in Japan mountains, to climb, and his goal in life was to climb all of them. He had climbed quite a number of them and had an accident ultimately. But before that, I got to see him speak in 1998, just before this paper was published, at a meeting in France. And he had this is a quote that I'm sort of paraphrasing. So he gave the keynote lecture because this work had just come out. And so let me describe the work first, and then I'll place the context of the quote. So what they did in Kinoshita's lab is they sort of contrived this experiment where they said, okay, we're going to put the F1 ATPase. You can run it in reverse by burning ATP. And so it usually makes ATP, but you can feed it ATP. It will burn it and run the reaction in reverse. That's what they were doing. And they said, well, what is going to be our molecular flea circus? What's our handle? In this case, it was going to be a fluorescently stained actin filament. Just happened at that time they were studying physical properties of actin filaments in their lab. So they had a lot of fluorescent rhodamine stained actin, okay? So they said, well, what if we had some way of actually sticking an actin filament onto the gamma subunit, and then we put it into a light microscope. If it rotates, we should just see it rotate, okay? And this is, in fact, his quote, that if I see one rotate, then by definition, it must rotate, okay? It sounds deep, right? It's not that deep, I guess. But it's true because in this experiment, many of his molecules did not rotate. Many of them were stuck to the surface in a funny way. They sort of agitated or they were hysterically hindered from doing their... But every once in a while, he would get ones that look like this. And so I'm going to play you a movie now. And what's interesting about this is that you can control the rate of rotation that they observe by how much fuel or ATP you feed the system. So this is a 10-fold greater concentration of ATP than here. So you're going to see that the rate of rotation here is much faster than what you observe here. So let me just play the movie. Right. So you'll see this one's rotating, okay? It's hard to argue with that. If it rotates, it must rotate, right? This one's interesting because you see it taking steps every third of a revolution. So it's sort of... It'll replay, I think, again. So it will sort of hang out, then step. And this is because it's at lower ATP concentration, so it's waiting for another ATP to come in. Finally, another one will come in. Well, it went backwards. That's another thing we learn in single molecule experiments, that these reactions are, in fact, reversible. Okay? And so you can see it taking a third of a revolution steps. And that also was a component of the model that was being tested, this rotation model. And again, this can be plotted as a function of time into the revolutions that they observe for individual complexes. So again, we're back to one of these type trajectories. But you can see here, for example, each individual revolution is nicely divided into 3 equal segments of 1/3 turn revolution. Okay? And so this is one of these instances where seeing is believing. For so many years, people debated, does this central subunit rotate? And this was settled at this point, okay? And they went on to publish a number of very exciting papers dissecting how the energy of ATP actually is coupled to this mechanical driving force, okay? There's a lot of really interesting stuff to be studied there. Here's another one that I really like, okay? So this is the process of vesicle transport, which is very dramatically animated here from, if you haven't watched it, I encourage you to Google the inner life of the cell. This is a lot of fun to watch. And what we're seeing here is an individual kinesin molecule, shown here, dragging its major cargo, the vesicle, right, as it's walking along its track, which is a microtubule in the case of a kinesin motor protein, okay? So this is kind of a dramatic image, right? You know, walking along. So how do we know that it's taking these individual steps? Well, the group of Steve Block and his student Mark Schnitzer at the time, they measured it directly using a technique called optical trapping, okay? Again, we're left with a squiggly line data trace, okay? But this time, notice as a function of time, we have some displacement in distance, nanometers, and notice how each step, so what we're looking at is an individual kinesin walking along a microtubule, and notice how each individual step takes 8 nanometers, okay? So it turns out that 8 nanometers is exactly the distance on a microtubule lattice where the contacts between the motor protein, just as you're seeing here, are 8 nanometers away from each other. So it would have been predicted, based on structural biology, static images, that 8 nanometers would make sense. Could have been 16 nanometers, could have been some multiple of that, right? And in fact, in certain cases, that is what is observed. But for the most part, you see these discrete 8 nanometer steps. You're watching a kinesin walk, okay? So we're allowed now to look at these movies and say, ah, yes, this is what a kinesin does. It walks along its track, okay? Another example of single molecule experiments sort of giving you a window into molecular behavior, okay? The last example that I'll show before talking about DNA some more is one of my favorite cellular machines, the ribosome, okay? So this is the protein translation machinery, again, being animated here in this movie. And what's happening is you're seeing a cross-section of the ribosome. This is the mRNA. tRNAs are flying in, and the nascent, newly synthesized polypeptide is emerging through the peptide channel of the ribosome. And you see, notice here as it's ratcheting along the mRNA, it's happening again in discrete steps. Now, we all learn about the genetic code. The genetic code is a triplet codon system of information. And so it would make sense that the ribosome would move like a train on a track in increments of 3 nucleotides along its mRNA substrate, right? That sort of makes sense. And indeed, as is being shown in this movie, if you set up a single molecule experiment, here what's being shown, again, just focus on the top plot here, as a function of time, is the extension here. And each of these black arrows indicates a step that corresponds to 3 nucleotides of the mRNA being pulled in to the active ribosome, okay? And so, again, you're seeing in real time a ribosome pull an mRNA into its active site to decode it into a protein. And you could imagine that this is just a starting point. So this is the observation of a live ribosome translating. And there are many interesting mechanistic questions that one could ask about how ribosomes work, how the machine actually functions. And this is a starting point because now you can study at the single molecule level that dynamic process, okay? So these are the machines that I think really provide a nice example of the power of single molecule observation. Okay, so that's about 25 minutes. So now I'm going to spend the rest of my time talking about research I've been involved in or supervised, looking now at what you might think is a much simpler system, just DNA. So I've shown you some very complex systems. You would think, and that's partly why we started working on this, that this should be easy, right? That's a very dangerous thought to have as a scientist. Nothing is easy, okay? And so, so what I'd like to describe first is some work that really got me into single molecule biophysics. This was a paper that my colleagues at Brno and I published in 2003. This was an image that we submitted but was not selected for the cover of the journal at the time that we published this paper. But I at least get to use this image for fun talks like this to share with you. And maybe you guys have played with one of these rubber band airplanes before, so where you twist the propeller and then you let it go and it flies away, right? So the nature of this experiment is going to be, well, what happens when we twist up on a piece of DNA, right? What can we learn about its physical properties by watching it untwist, okay? This is the inspiration behind this funny cartoon, okay? So I start this little component of the talk with this quote. So inevitably, as biochemists, structural biologists, whatever one wants to call oneself, we will inevitably use the term structure-function relationship, right? The structure-function relationship of such and such molecule, right? And I like to realize that that notion of structure and function in this really intimate relationship between the two is actually borrowed, in a sense, from the area of architecture, right? So Louis Sullivan, in the late 1800s, first coined the phrase, form ever follows function. And this is a design principle in architecture, right? And that you can, it's very important to think about the form of a structure when you're thinking about what the function of that, in the case of architecture, a building or a living space, whatever that might be. So this holds true for molecules as well. And I think the best example, or simplest example perhaps, is the double helix itself. So the DNA double helix, when it was first reported in 1953, now, I don't know if you guys do it, but on our campus, April 25th is DNA Day. We all dress up as different bases, and we do funny dances in the form of DNA in the fields. And so we celebrate this day as being sort of a demarcation point. There was sort of the time before we understood the structure of DNA and the time since. And this really typically marks the revolution of molecular biology, is this day in 1953. And, Watson and Crick, concluded their very short paper describing the model of the double helix that ultimately proved correct with this very, prescient, but at the same time slightly, I don't know, the word is escaping me, but let's just say thoughtful observation that it had not escaped their notice, right, that the structure that they proposed, this double helix, immediately suggests a mechanism for how the genetic material might be copied, okay? So this is a very clear example of form following function, or function following form, let's say, right? Because as soon as they realized that you had this sort of anti-parallel complementarity of the two strands of DNA in a double helix, it occurred to them that if you peel these two things apart, you now have templates to make two new copies, okay? We now know that that's exactly how DNA is replicated through a process drawn here as so-called semi-conservative DNA replication, where each strand in the parental molecule serves as a template to synthesize its complementary strand in each subsequent daughter molecule. So this is a classic example of how learning about the structure or modeling the structure immediately makes one think, wow, maybe this suggests how the genetic material is copied, the mechanism for this. Of course, the machinery, the polymerases, the helicases, the primases, none of that, they had no knowledge of that. This is purely conceptual at this point, right? We now know a lot more, okay? But again, their model and this image was a static image. So I posed the question, but well, what about the dynamic properties of DNA? Okay, it's not enough to just think about a static B-form DNA helix. And in particular, what about the mechanical properties of DNA? So what do I mean by that? So it's important to recognize, and this sort of goes back to this image, this artistic rendition of David Goodsell that I put forth, that DNA is highly contorted inside the cell. It's subject to all sorts of torture, okay? And so I put up here, this is another sort of analogy or metaphor that is losing its efficacy because newspapers are not so popular as they once were. But this is a printing press, okay? And if you imagine that the paper going through the newspaper printing press is the DNA, okay? It's being sort of rapidly shuttled and pulled and wound around. Well, the DNA, without focusing too much on all the individual machineries, it's essentially undergoing the same thing every time it's being copied, okay? So there's all sorts of machinery that the DNA is acted upon by. And this machinery spins the DNA, twists the DNA, bends the DNA. And so it's actually very pertinent to ask, well, how does the DNA as a material respond to all these mechanical perturbations? Because that's going to relate to how it's able to pass through all this molecular machinery. The other thing to consider is that DNA is a very thin molecule, of course, 2 nanometers across in the diameter, but very long molecule, okay? So all this DNA has to be compacted and confined to a very small, relatively small volume, okay? So in the case of naked DNA, this is just electron micrographs of plasmid DNA. Sometimes that compaction takes the form of what we call just plain supercoiling, twisting of the DNA about itself. That's what you're seeing here. This is a relaxed plasmid, a highly supercoiled DNA, which is occupying less volume. But of course, in higher eukaryotic cells, we know that the compaction process of DNA begins at the level of wrapping the DNA around these nucleosome core particles, and then there's this sort of hierarchical organization of chromatin, let's say, that achieves compaction. And it's one thing to say, okay, well, you twist it up or you wrap it around, and that compacts the DNA. But it's actually informative to do a very simple back-of-the-envelope calculation, how much compaction actually has to happen for the DNA to fit inside a cell, okay? So we can do that for a bacterium, and we can say for a single E. coli cell, which typically is thought of as being a 1 micron or so across vessel, right, in diameter, you have just under 5 million base pairs of DNA. We know the crystallographic unit of each base pair is 0.34 nanometers. So it's actually quite easy to calculate how long the DNA molecule is from an E. coli cell, okay? And it turns out to be 1.5 millimeters or so, end to end, if you were to stretch it out, okay? 1.5 millimeters that has to fit into essentially 1 micron cube volume, okay? And I say that's kind of impressive. But you guys are like, nah, that's not that impressive, okay? But, okay, let's talk about you and I, okay? So you and I have, in each of our cells, about 6 billion base pairs in our genome, obviously divided into 23 chromosomes from mom, 23 from dad, okay? You add these up together, 6 billion base pairs. If you now do the same calculation, each of our cells, DNA, end to end, okay? Now we're talking about is 2 meters long. 1 cell's worth of DNA, 2 meters long, okay? That's a lot of DNA in size, okay? Our body, this is actually, I put this number 1 trillion cells. It's actually a very controversial number, but we'll assume it's 1 trillion for the moment, okay? So we have about 1 trillion human cells in our body. You have even more bacterial cells in your body, but that's a discussion for another day, okay? But if we just count our human content, okay, and we add up all 2 meters from all trillion cells, now laid end to end, there's enough DNA to travel from Earth to the sun and back about 6 times, okay, in our bodies, okay? So this hopefully conveys to you, and I say this is definitely impressive, at least in my opinion, okay? And so how does all this DNA fit into the nucleus of a cell? And even more perplexing, when the DNA needs to be accessed, say to be transcribed, how does the transcriptional machinery find, right, in this sea of DNA, the appropriate site, okay? So all of this is to say that understanding how DNA is a material, okay, a polymer, bends and twists, is very important in understanding a process such as this high degree of compaction, okay? So in the earliest days, I always make the joke, when my son was young, like a toddler, you know, his approach to understanding the world is very similar to mine is right now in the laboratory, which is if you want to understand how something works, you break it, right? You sort of pull it apart, right? And so if he would, when he was 1 or 2 years old, he would pick something up, pull on it, right? That was sort of his natural instinct, right? I'm still doing that at age 42 today. And in fact, some of the earliest DNA micromanipulation experiments shown here were nothing more than that, right? Which was, let me take a piece of DNA and pull on it, okay? And ask, how does the DNA respond to that pulling force, okay? So that's what we're looking at here for double-stranded DNA, shown here, and single-stranded DNA, shown here, okay? This is the extension, normalized extension, as a function of force, which is on these scales of nanometer distances, the relevant unit of force tends to be piconewton forces, okay? So without going into too much detail, you can immediately see that double-stranded DNA has a very different elastic response than single-stranded DNA. Would you agree with me there? Yes? I'd like to see a few heads nodding, making sure I have you still with me. And so these early experiments first characterized unique structural properties of double-stranded versus single-stranded DNA. There were some very unexpected structural transitions. You can see when you get up to about 65 piconewtons, something very strange happens to double-stranded DNA. I won't go into too much detail there. But there were, again, these were the surprises. Nobody anticipated that when you pulled DNA in a certain way, it would suddenly go to much longer than its original crystallographic length. But I would say perhaps even more important than some of these polymer physics type experiments was this point here, which is that there are many processes inside the cell. If you're talking about replication, taking a single-strand template, turning it into a duplex product. If you're talking about unwinding of DNA by a DNA helicase motor, taking a double-stranded DNA and turning it into a single-strand product, you could see that basically the single-stranded and the double-stranded DNAs at a given force had very different elastic responses. And so researchers were able to utilize this framework, right, this knowledge of the underlying behavior of DNA, to devise assays to follow replication in real time. Watch the conversion of duplex DNA into single-strand DNA, for example. So I think that's perhaps one of the most important points, is that these basic physical measurements really lay a platform for biological mechanistic type experiments, okay? So this was in 1996, it was published. When I began work in this area, we were interested in doing the comparable experiment, but rather than pulling on it, which was relatively easy to do, we wanted to twist on the DNA. And this was an experiment that my graduate advisor had been trying to convince students for a decade to try to do, okay? And I was the one who was naive enough to actually say, sure, I'll do that. Why not? Doesn't sound so hard, you know? But we managed to figure it out. And so I just thought I would show you an example of what this experiment looked like. So we were using optical trapping technology at that time. That's, again, here's our molecular flea circus. We can hold on to that bead in a laser trap, okay? And the other end of the bead was attached at this time to a glass capillary. And we would attach a single piece of DNA between these two beads. So that's our handle. And that's, in fact, how the DNA was stretched in those earlier experiments, okay? But in this experimental design, it was a little bit more involved because we wanted to track the torque response of the DNA. So in order to do that, what we did was we engineered in what's called a single-strand nick. This is a single break in the phosphodiester backbone of DNA, which introduces a free swivel into the system, okay? And then just above that, we modified the DNA so we could put what we call a rotor bead, which was basically a fiducial marker for the rotation of the DNA, and we could stick that to the side of the DNA. So you could imagine, it's easy to draw this in a cartoon. To assemble this as an actual experiment is a little bit of a different story. This entire experiment is being conducted in a small microscope chamber. In fact, many of you who are doing the practical, we will be playing with these chambers over the course of the next couple of days, all done in aqueous buffer, okay? And one of the advantages of doing it in aqueous buffer is that we had control of the hydrodynamic flow, right? We could pass buffer across the system. And so what we were able to do is we would apply a flow, kind of like a current, right, of a river. And that current would hold the bead downstream, hold it fixed. And while we were holding it fixed there, we could rotate the pipette, introduce torque into this upper segment of the molecule. And when we were ready, we would then stop rotating the pipette, turn off the current, and see what happens to that rotor bead, okay? So here's what the actual experiment looks like. So this is the laser trap. You can't see the laser, but it's there, okay? That's what's holding this bead. Here's the glass capillary, second bead. There, again, you can't see the DNA, but it's there, okay, between the two beads. And here's our small 500 nanometer diameter rotor bead, okay? At this point, what I had done is already introduced several hundred turns into the system. And when I turned the flow off, again, you could tell I was inspired by Kinoshita's rotation experiment, because we said, well, if it's rotating, it must rotate, right? Okay? And what we had created here, to our knowledge, was the world's smallest wind-up toy, okay? And so we had wound it up, and now the DNA is powering, right? The torque accumulated in that upper segment of the DNA is powering the revolution, or the rotation, of this rotor bead. And so as this is rotating, you can watch that and sort of see what you observe, okay? And what we could do is we could track that in this type of plot here called the kymograph, where we just take a section of this bead. And you can see that basically right when we turn the flow on, you see it's slowing down now, so the energy has dissipated from the molecule. And you can see that perhaps better here, where you stack these images over time, and you can see that it forms, interestingly enough, a helix in time, this rotation, okay? And then eventually, you see the pitch of that helix is pretty uniform, and then it begins to slow down and dissipate, right? So what we had done is we had wound up the molecule, and then we had watched it unwind, okay? So we can take that information, and we can ask, well, what was happening to the DNA during that experiment? So what's plotted here is now that angular velocity, the rate of the rotation that you were just observing, again, as a function of time. And if you just focus on the green plot here, you can see that for a long time, the angular velocity is constant, okay, until finally it decays. And it decays down now just to Brownian fluctuation. It's just sort of bouncing around from thermal noise. In the interest of time, I'm going to sort of forego some of the structural conclusions. But one thing that you can do here is each individual measurement is quite noisy, right? But you can do many such measurements, okay, as shown here. And if it's in fact really just noise, then it should average out if it's just random Brownian noise. And so this is about 30 or 40 individual trials of the experiment shown here, overlaid, right? And this is the simple equation. One of the things that I love about single molecule biophysics is that the math that one needs to use to understand some of these results is quite simple, actually, which is good for people like me, trained in biology. And so what this expression tells us is that the torque, the instantaneous torque, at any point along this experiment is related essentially to the angular velocity omega, shown here, and then the rest of these terms, it goes as the cube of the radius of the bead, so on, and the viscosity is eta here. So essentially what this tells us is that we can take our observable, the angular velocity, and convert this directly to the torque being output by the DNA at any point in time in the experiment. And so that allows us now to get out of this angular velocity regime and into a torque regime. And so now what we could do is average all these trials. We could overwind the DNA, underwind the DNA, depending on which way you're spinning it. And you could study some really interesting transitions, very similar to what had been done with the stretching experiments that I started out by describing. And so, but I think the most important finding is what happens here in this approximately linear response regime, okay? So it's in this regime, so here at this intercept is where the DNA is completely relaxed, okay? As you're moving up this curve, you're overwinding the DNA. And as you're moving down this curve, you're twisting it in the opposite direction. And what you can see here is that across a fairly wide range of torques, DNA behaves as a torsional linear spring. That is to say that the more you twist it, the greater the torque is, right? We all learn Hooke's law, for example. So this is the twist equivalent of Hooke's law, okay? And so the larger the angular displacement, the torque increases linearly. And directly from that slope, you can extract the so-called torsional modulus, or the C constant of DNA. And this is a constant of the genetic material that tells us something about its fundamental physical ability to absorb twist and generate torque. And that's going to tell us about at what point the molecule buckles and how it's able to compact. And when people simulate compaction of DNA or physics of DNA, this is a very important parameter that goes into such simulations. And it turns out that when that number had obviously been measured many times before we measured it, but using a variety of assumptions. When you do a simple single molecule experiment like this, using, it's simple now, right? You just get the number, right, directly out. And it turned out that the torsional modulus that we measured of about 410 was 40% higher than all of the accepted values in the literature, okay? So I'm proud of this measurement because it's a relatively simple experiment. We measured a number. There's more, but we essentially measured a number. And this paper is now very highly cited. It's my most highly cited paper because anybody who models DNA needs this number. And people like our number because it was a very simple experiment, okay? Great. Okay, so that leaves me with about 10 minutes left. And I promised I would talk about FRET, so I'm going to talk about FRET now, okay? So here's an experiment done by one of my former graduate students, now a postdoc at the HHMI, Howard Hughes Medical Institute campus, Janelia Farms in the States in Maryland. Selena, looking very happy here. She's outside of the lab, you can see. So that's good. She's happy in the lab as well. So it's possible to be both. And what she was interested, so this is interesting for me because having come from these early days of DNA structural studies, as Stirad mentioned, my laboratory is very interested in, again, DNA structure, but now not just any old DNA. You might wonder, what DNA were we pulling on in that experiment? And I'll tell you about that later. We didn't really care at that point. We weren't so concerned with what the nature of the DNA is. But now, it's 15 years later, we want to study specific DNA, and specifically DNA found at what's called the telomeres in our genomes, okay? So I'm sure many of you are familiar with the notion of telomeres. Telomeres, as shown here in a fish image, a fluorescence in situ hybridization image of a metaphase spread, can be sort of depicted with these fish probes as these little yellow dots capping off the ends of the chromosomes. And so there are these unique DNA sequences found at the ends of linear genomes. These are telomere DNA sequences. And I could give an entire talk, of course, on telomere biology and function, but I'm going to boil that down to 3 bullet points here in the interest of time. Telomeres are critical to our genomic stability because they essentially protect the ends of the genome from unwanted recognition or from DNA damage response machinery or other processing machineries, okay? And so they basically tell the cell, I'm a natural end, not a DNA break. Leave me alone, okay? Now, there's a big question as to how they achieve this, very important function. And these are the sorts of questions that people like Stirad and myself spend our nights awake wondering how that actually works. As you may also know, I didn't go into detail, but as our genome replicates with each round of cell division, because of the nature of the replication process, the DNA genome gets shorter and shorter in the absence of some mechanism to maintain genome length. This is often referred to as the end replication problem. It's not necessarily so much of a problem because that shortening essentially creates a finite lifespan for any particular cell. And so the telomeres shorten, and when a telomere gets critically short, the ability to preserve genome stability may become compromised, and the cell is aware of this in some magical way and stops growing, okay, in a controlled manner, undergoes so-called cellular senescence, okay? And so any cell that's rapidly dividing, such as a pluripotent stem cell in our body that hasn't differentiated yet into whatever type of cell it's going to become, needs to somehow counteract this shortening growth limitation. And it turns out that in a disease context, the vast majority of cancers that have been studied also have come with some mechanism to maintain telomere length. And that typically is the enzyme telomerase, which I'm not going to talk too much about today. But that's essentially the enzyme machinery that maintains telomere length. But I want to talk about the actual DNA found at telomeres. So our telomere, you and I have telomere sequences that look like this with a hexameric or 6-base repeat, TTA-GGG, TTA-GGG repeating, okay? Much of the telomere is duplex, so this would wrap around the Mendel Museum here, okay, if it was drawn to scale. So on the order of, let's say, 10 kilobases of this repeating unit of DNA. But then all of our telomeres are processed to have a single-strand tail which is, again, of this TTA-GGG sequence. We're going to focus on this single-strand tail for the next 5 minutes as I finish up. Okay, so let's focus on this tail. So it turns out that sequences that have these repetitive runs of guanine can form an unusual structure, which had long been appreciated. And that structure is based in the ability of guanines to form these so-called quartet, planar quartet structures, where not only this is the Watson-Crick face of the base, and it's interacting here with a different side of the guanine base called the Hoogstein side, named for Professor Hoogstein, who first described this type of base pairing interaction. And so you can have a combination of these Hoogstein base pairing interactions to form a planar G quartet, it's called. And these, for those of you who will participate in the practical this afternoon and tomorrow, we're going to study this process right here by which a single monovalent cation is capable of being coordinated inside this quartet structure, and depending on the size of that cation, is better or worse at stabilizing such a structure. And I don't want to give away our experiment, but we'll study different identity cations. Now, when you have these stretches, you can form a myriad of structures. Again, these are the identical sequences. I just show these structures to give you a sense of the diversity of structures that these can fold into. In blue are shown these stacked quartets. So you can see that the quartets are preserved in each case, but the topology of the backbone, the connectivity, is unique between each of these structures. And the key point that I'll have you note is that these are all the identical sequence. Okay? They're the same sequence forming into different structures. People are very interested in these so-called G quadruplex structures because quadruplexes have been long known to inhibit the enzyme telomerase and also potentially disrupt telomere-protein interactions and normal telomere homeostasis. So there's an abundant literature of small molecule ligands, drugs, potential drugs, designed or being developed to target quadruplexes in the interest of being used as anti-cancer therapeutics, okay? And none have succeeded yet. And I would argue that part of the reason that none have succeeded is that we don't fully understand the nature of these quadruplex structures, okay? And that's part of what motivates our work. So just very briefly, FRET, we'll talk more about this during the practical, but it's important for me to note that the notion of FRET, wherein a donor fluorophore and an acceptor fluorophore interact with one another through a dipole-dipole type interaction. You can directly excite a donor, and if that donor is in sufficiently close proximity to an acceptor, you can get what's called the non-radiative energy transfer between the donor dye and the acceptor dye, and you can get emission from an indirectly excited acceptor dye, okay? This is not a new idea. In fact, Stryer and Haugland, you may know Stryer from the famous biochemistry textbook, Stryer, in the late 1960s first described the principles experimentally of FRET, and they showed this very sensitive dependence on the distance between the two dyes on the efficiency of the energy transfer process. And so we can use this sort of sensitive dynamic range to study relative positions of FRET dyes in a particular experiment. But it took, like about 30 years before Tak Jip-ha, who is a leader in the field of single molecule FRET, while he was a student, to demonstrate that you could detect single DNA molecules labeled with FRET probes. That's what's being shown here. And then in a dynamics measurement in the early 2000s, my postdoctoral advisor, Xiaowei Zhuang, was one of the early people to show for a small, simple RNA docking-undocking transitions, dynamics of an RNA molecule using the FRET technique, okay? We're going to do an experiment very similar to this one during the lab practical. So we'll talk more about this tomorrow. This is what a FRET microscope looks like. You can get some interesting data. In the interest of time, I'm going to skip over that for now and just tell you about one last result before I conclude. So in this particular experiment, what Selena was interested in doing is studying one of these quadruplex structures. But she was interested, again, in applying tension to that quadruplex structure and asking, how does its folding dynamics change as a function of tension? And the way she set out to do that was actually which is a common sort of theme in single molecule science now. When I started in single molecule biophysics, if you would go to a meeting, the fluorescence people would sit on this side of the aisle, and the force spectroscopists would sit on this side of the aisle, and never the two shall meet, right? Now it's not so bad. We're a couple of generations outbred from the early pioneers, so we can talk to each other and, in fact, do the same type of measurement in a single observation. So that's what's going on here, where we use a technique called magnetic tweezers to pull on the DNA molecule, and we use fluorescence to read out what's happening locally at high resolution. Okay? So this is what the experiment looks like. This is exactly the experiment that we'll do in the laboratory here in Brno, where we first wanted to explore, well, what is the behavior of the quadruplex in the absence of any pulling force? And so you can see here, this is a dye intensity trajectory. This is the calculated fret value. All I really want you to get from this, again, we're looking as a function of time. We're looking at dynamics. And you can see that even in the absence of any structure, just going to leave that down there for now, is sort of a change in structure. Okay? And this is a great system to study with students. It's very reproducible, and it happens on relatively long time scale. You see this is 2 minutes long movies, and it spends some time, maybe 30 seconds, and it unfolds, switches to a new structure. Okay? So we'll take a look at that more tomorrow, or this afternoon as well. But then the question is, well, what happens if I begin to apply some perturbation, some tension along a structure? And you can immediately see, so here is the low force experiment. Here is the high force experiment. The higher state here is the folded state, okay, as shown here. The lower force is the unfolded state, okay? And what you can, or the lower fret is the unfolded state. And you can see as you pull harder, as your intuition would tell you, you spend more time in the unfolded state, right? These times down here in the low fret regime are becoming longer. Yes? Okay? So from studying the force dependence of the time spent in the folded and unfolded state, you can actually extract some very interesting things about the energy landscape of this folding-unfolding process. So this is my last data slide, and then I'll conclude. So you can use these expressions here, which in your chemistry classes you learn about Arrhenius dependence of a chemical reaction, and how chemical reactions depend exponentially on the temperature, right, for example, of the reaction. And so we can take a similar type of transition state theory and apply them to experiments in force spectroscopy, shown here, where we can say for any, here I'm showing the dwell time tau at no force, that basically the amount of that dwell time, the value of that dwell time is going to vary exponentially on the amount of force that you're applying. So it's very sensitive to the force that you're applying. Multiplied by this interesting term here called the characteristic distance to the transition state. So let me just explain for a moment what that is. So if we imagine an energy landscape shown here, unlike most energy landscapes that you might have learned about or studied in chemistry classes, where you have free energy as the function of, this is usually labeled reaction coordinate, but the reaction coordinate is not typically very well defined. Okay? In a force spectroscopy experiment, one of the great advantages is that that reaction coordinate is very well defined because you're pulling along the reaction coordinate. So we can define that reaction coordinate in a true unit of nanometers, okay? And so when we extract out the distance, we can say, well, from the relationship of the time spent, say, in the unfolded state as a function of force, we can get information about how far, basically, it is from the folded state. So if we imagine that we're pulling it apart, right? How far is it basically from that folded state to the transition state of the unfolding reaction? That's the type of information that we can get out. We can fit these sorts of data to these expressions to get these numbers. The data are well fit by this simple model. And this is the picture that emerges, okay? And so what turns out is that for a folded quadruplex, when you pull on it, the distance to the transition state is very small. It's about 0.5 nanometer, okay? That number doesn't mean very much to you, but the fact that it's very small is actually fairly surprising. What that means is that we typically call G quadruplexes alternative secondary structures of DNA. But in fact, small distances to the transition state for unfolding is more characteristic of something like a protein that is stabilized by tertiary contacts. And so that was a little bit of a surprise to us. And what it really tells us from the perspective of the behavior of quadruplexes is that once you begin to peel off a few of these bases from a G quartet, the stability of that structure is lost and it very rapidly unfolds. And that was somewhat of a surprise because the sort of calorimetry-based methods for studying these structures had suggested that these structures, rightfully so, are very thermodynamically stable. So TMs of on the order of 85 Celsius, right? Very stable structures. So it came as a surprise that a small mechanical perturbation makes the structure completely fall apart. And the reason for that is that that mechanical perturbation is happening at a very specific location of the structure, whereas when you heat up the system, basically all the degrees of freedom of increasing the temperature of the system are distributed across the entire structure. And that has really important implications for an enzyme that unwinds quadruplexes and the sort of task that it must achieve, as well as if you were going to design a G-quadruplex stabilizing drug, you might want to understand what part of the structure is best to target in order to interfere with biological processes. So that's just an example, again, of something that we've learned about G-quadruplex DNA. And now we're studying things like proteins and enzymes that unwind these structures. So I conclude with this. So I realize I'm in Europe. Baseball is not so big here. In the States, baseball is my favorite sport. The Yankees, most of you, I think, have heard of the New York Yankees. Yeah, okay. If you've heard of one team, it's the New York Yankees. It's my favorite team. They just lost last week in a big game, so their season is over. But the late Yogi Berra, who is a famous player from the early days of the Yankees, was famous not only for being a very good athlete, but he said some very funny but also very wise things. And one of the things that he said, one of his famous quotes is, "You can see a lot by observing." Right? So he was famous for saying these kind of silly things. But it's very apropos, right? So here, if you can, this is really the motivation behind this entire workshop, right? Is that if you can design an experiment to see it, right, you can immediately learn quite a lot. So who would have thought we could learn such a lesson from an old-time baseball player, okay? So with that, I'll just conclude. I obviously have funding, mostly from the National Institutes of Health, various people in the lab, and a bunch of great collaborators. And I'll just point out that my laboratory, in addition to having fun doing science, we compete in our annual costume contest every Halloween. You might recognize this as Monty Python and the Search for the Holy Grail here. We won this particular year, so we're very proud. So thank you very much for your attention.